Hello, and thanks for joining us today for the latest Technology Networks webinar, Everyday Culture Practice, Improving Reproducibility in Cell Culture. I'm Anna, Editor for Technology Networks, and I'm here to moderate the event. I'm pleased to have Dr. Jessica Wagner joining us today as your presenter. Dr. Wagner is an application specialist focusing on cell handling at Eppendorf headquarters in Hamburg, Germany. Entering the cell culture laboratory as a graduate student for the first time in 2006, she came to Eppendorf with over 5 years of experience in cell culture. After studying biology at the universities of Marburg and Dusseldorf, she investigated apoptosis mechanisms in different cell lines for her diploma thesis. Following 1 year as a research associate working with primary cells, she started to work towards her PhD with a focus on reproductive biology at the Medical School Essen. She joined Eppendorf in April 2012, shortly after completing her PhD in biology. A warm welcome to you, Jessica. Following the webinar, we'll have a Q&A session, and we would welcome any questions that you may have. To ask a question, you should enter your question into the box on the left-hand side and click Submit. We will answer as many as possible during the time available. Any questions that we don't have time to get to, we'll be sure you are contacted offline with an answer. If you should experience any technical issues, please use the question box to request support. And also, please remember, you can ask questions at any time, even whilst watching the webinar on demand. I'll now hand over to Jessica.
Thank you, Anna, for the introduction. Hello, everybody from my side, and welcome to our webinar. After a short introduction to the topic, I will talk about some general aspects that influence reproducibility of cell culture experiments. We will then have a look at the routine culture steps and outline the best methods to improve overall cell quality. In the end, we will touch the topic of misidentified cell lines and give some advice for cell authentication. I want to start with showing you some results of a current survey conducted by Nature magazine in 2016. In this survey, more than 1,500 researchers of all different scientific disciplines have been asked if they see a current crisis in reproducibility. As you see here, more than 50% of researchers surveyed agreed that there is a significant crisis, and taken together, more than 90% do see reproducibility as a slight to significant problem in science. Shown here on the right-hand side of the slide are the responses to the question if there are any measures undertaken to enhance reproducibility. And although the topic seems to be on people's minds when you have a look at the results of the first question here on the left, still more than 1/3 of the researchers have never established any procedures of reproducibility in their lab. No matter which scientific field you look at, there are a few general principles that should be followed in any scientific process. A rigorous experimental design, including proper statistics for robust and unbiased results. A clear and complete documentation of both the experimental procedures and the obtained results. Transparency in reporting the results, as well as experimental details, and the reproduction of results are cornerstones of good scientific practice. Using cultured cells as model system has indisputable benefits. Especially primary cells retain many functions and properties of their parental tissue, which make them an ideal model system to test new therapeutic approaches. From initial target validation studies through clinical candidate selection to subsequent translational studies, the use of continuous cell lines as a renewable resource of cell material is essential across numerous areas of biomedical research. But the cultivation of eukaryotic cells has some major drawbacks or risks that, when overlooked, can lead to severe problems in reproducing experimental data. I think everyone working in a cell culture lab is aware of the risk of having contaminations with microorganisms that overgrow and ruin the cultures. Especially Mycoplasma are difficult to detect and therefore a special threat in cell culture labs worldwide. Cross-contaminations of different cell lines, which I call eukaryotic contaminations, are a widespread problem that endangers the reliability and reproducibility of experimental data. Besides microbial and eukaryotic contamination, it is often overlooked that continuous cell lines, which have an unlimited lifespan, can undergo genetical changes. So although the cells continue to proliferate, their genotype as well as their phenotype can change over time. The cultivation conditions can have great effects on the cells. And here we are not only talking about specific experimental conditions that result in different cellular responses. It's about media formulation, the use of serum, or such simple things as incubator door openings. Cells can respond to the most subtle influence. All these risks and drawbacks need to be managed for cell culture experiments to be reproducible. And with this webinar, I want to give you some practical tips how to incorporate certain standards into your cell culture routine that help to improve reproducibility. But before we proceed, here comes our first poll questions. As you see, it's a very general question, and I'm curious about your answers. Do you think that cell culture experiments are more difficult to reproduce than others? And the answers you can click on is yes, no, or I don't know. So I'm waiting a few seconds until your answers are coming in. And here now I will show the results to you. So, okay, so we see that more than 50%, almost 60% of you do see or do think that cell culture experiments are more difficult to reproduce. Only 26% said no, and 15% is not sure about it. So let's have a look at the steps that are usually part of the cell culture workflow, independent of the specific cell type you are working with. I want to start with the two steps here on the right, seeding of cells that are used for an experiment. I won't go into any specific type of experiment, but rather talk about some general aspects that should be considered. This is what we are looking at in the next chapter, how to ensure best experimental conditions. So let's have a closer look at this first experimental step that is so important, the seeding of cells. The precondition for getting comparable results is to seed out the same number of cells for every negative control, every positive control, and for every experimental replicate. The more replicates you include, the better for your statistical analysis, of course. Variations in seeding cell numbers and the formation of air bubbles during seeding will not only increase standard deviations, it will also make your results less reliable. Repeated mixing of cell suspension during seeding and careful pipetting can be a solution. Let's go more into the detail on the next slide. When you fill, for instance, a multi-well plate with cells, it will take some time until you have filled all wells. You can use a single-channel pipette and have the cell suspension in a tube, or you can use a multi-channel pipette and have the cell suspension in some type of reservoir. No matter which tool you use, the longer the seeding process, the higher is the variation in cell number from one well to the other. As you see in the image here on the upper right, the cells sink towards the bottom of the tube with time, and this sedimentation goes quite fast, within minutes. Therefore, you need to resuspend or mix the cells to swirl them up, and in between to get an equal number of cells in every well. Saying that, we should keep in mind that cell culture media tend to foam due to the high protein content of supplemented serum. Air bubbles can hinder cell attachment. In the lower image here shown on the right, you can see how an air bubble hinders the cells nearby to adhere properly to the surface. So you should avoid harsh and fast pipetting to reduce the formation of air bubbles and also to reduce shear forces, which further stresses the cells. In fact, there are a lot of aspects about pipetting that are often underestimated. Pipetting is an everyday task and can have a huge impact on cell culture experiments. So first, let's talk about the different pipetting systems. Here you see how a classical pipette works. It is a so-called air cushion system. By pushing the control button, you push down the air above the liquid, and the liquid gets released from the tip. With this system, you have variable influencing factors, which I will talk about in the next few slides. They are best suitable for aqueous solution, and if you do not work with filter tips, the inside of the pipette can be contaminated by aerosols. Positive displacement systems, which you probably know as dispenser or stepper, function without air cushion. The piston is part of the pipette tips and is in direct contact with the liquid that is pushed down and released. So these pipetting systems are contamination-free and are unaffected by the type of liquid you are pipetting. And by the way, as there is no air inside the dispenser tip, you cannot create any air bubbles during pipetting. I already talked about pipetting different liquids. When you treat cells with any recombinant proteins or inhibitors, these are often supplied as lyophilized form and need to be dissolved before use. Commonly used dissolvents are alcoholic solutions like ethanol as well as DMSO. Both liquids can be difficult to pipette. Ethanol is a volatile liquid that tends to drip out of the pipette tip. With DMSO, you have a quite viscous liquid. So what some people do is they cut the tip of the pipette tip to create a wider opening. In both cases, alcoholic solution that is dripping out of the pipette tip and a tip that is cut to create a wider opening for viscous liquids, you cannot pipette an exact volume. But especially when pipetting any critical proteins, drugs, or inhibitors that you want to test with your cells, exact volume is essential. Just imagine you want to test different concentration of substance X, and you cannot be sure if the volume you pipette is precise. So looking at the different pipetting systems I talked about in the previous slide, a dispenser is the better choice here, because its precision is unaffected by the type of liquid you are pipetting. Not only the type of liquid influences the pipetting results, also the pipetting technique plays a role, especially when using air cushion pipettes. Hold the pipette vertical when aspirating the liquid, as seen here on the left image. Immerse the pipette tip only slightly and try to keep a constant immersion depth. If you follow this technique, you will improve the precision of the volume that you are pipetting. Now let's have a look of how pipetting can affect a cell-based assay. In this graph on the left, you see the results of a cell viability assay. Each column represents one row of a 96-well plate. The aim was to seed an identical number of cells in each well. 2 rows were left out for blanks. The cells were seeded with a manual 8-channel pipette from a reservoir. After 24 hours, the number of viable cells was determined using a colorimetric assay. In rows 1 to 5, you see varying cell numbers in the wells and therefore high standard deviations. Here the user did not mix the cell suspension between pipetting each row. So the cells sedimented in the reservoir with time. In addition, the pipette was held in different angles and heights during aspiration. In rows 6 to 10, you see the results with mixing the remaining cells before pipetting each row, aspirating at constant height and in upright position. Taken together, these data show that the pipetting technique has a big influence on the results. Besides the technique and the pipetting tool, the other thing that can affect results of an assay are the so-called plate position effects. If you work with a 96-well plate, the usual volume you use is 100 to 200 microliters per well. Especially during longer incubation periods, evaporation can become a critical factor with this small volume, especially in the outer wells. Because here the wells are not surrounded by other wells with warm liquid inside, the evaporation becomes more severe. This is a phenomenon that we call the edge effect. So the outer wells of the plate cannot be used because the evaporation leads to different cellular responses. What you can do to avoid this edge effect is to fill the outer wells only with medium or PBS to insulate the inner wells. If you do that, you can avoid the edge effect, but you reduce the number of usable wells to 60, which is a loss in capacity of 38% per plate. The Eppendorf cell culture plate has a surrounding mold that can be filled with liquid. Thus, the outer wells get insulated and the evaporation effects are minimized. And here we measured how effectively the edge effect is reduced in the Eppendorf plate. Without insulation, the evaporation in standard cell culture plates can go up to 15, almost 20% in the outer wells. And again, if you think about a volume of 100 to 200 microliter per well, and you want to test maybe different concentration of a substance, Over time, the evaporation will influence the concentration of the substance, and the cellular responses are not comparable anymore. So with insulating the outer walls, you ensure stable conditions throughout the complete plate. In addition to stable conditions throughout the plate, the conditions for the cells in the CO2 incubator should be as stable as possible. Every time the door of the incubator is opened, the atmosphere inside is disturbed. Temperature, humidity, and CO2 level all have an influence on the cells. It is a good advice to reduce the traffic in and out of the incubator as much as possible. Only open the door when necessary and close it as soon as possible. An incubator with split inner doors helps to keep the atmosphere stable because the area that is opened is smaller. Especially when you work with very sensitive cells or when you work in a lab where many people are sharing an incubator, it can be a good idea to have separate incubators for maintenance cultures and running experiments. So you seed the cells for an experiment, do the treatment, manipulation, or whatever you want to do, and then put the cells into a separate incubator for the time of the experiment. Your colleague who has to split his cells or change the medium uses the other incubator, and thus your experiment is not disturbed by fluctuations of temperature, humidity, CO2 level, and maybe also oxygen level if you work under hypoxic conditions. So again, here is an overview of the general cell culture workflow. In the next chapter, I want to talk about some aspects you should consider to improve the overall quality of your cells, because not only the experiments itself plays a role when it comes to reproducibility, also the routine steps like freezing and passaging and the cultivation can be a critical factor. But before we go on, I would like to start the next poll question. I'm interested in the cell types you are working with. So the answers you can click on is continuous cell lines, primary cells, or both. And again, I will wait a couple of seconds until you clicked on the answers and until the answers are coming in. So thank you for participating. I'm now showing the results to you. Okay, so we see that 44% of you is working with continuous cell lines, only 14% with primary cells, and 42% is using both continuous cell lines as well as primary cells. So no matter which cell types you are working with, there are some ways to optimize the routine culture steps to improve the overall quality of your cells. I want to start with the cryopreservation of cells. During freezing and thawing of cells, the stress factors should be minimized. When you freeze the cells, you should be quick from the point where you add the DMSO containing freezing medium to the cells until you start to cool them down. The cooling procedure itself should then be slow to avoid the formation of ice crystals. To achieve a cooling rate of -1 degree per minute, use a special freezing container and put the cells into a -80 freezer for 24 hours. You should not store the cells for longer time at -80 degrees, but freeze them in a liquid nitrogen tank. When you thaw the cells, time is again critical because you want to minimize the exposure to cryoprotectants like DMSO. So the cells should be thawed as rapidly as possible and then be diluted to decrease the concentration of DMSO in the medium immediately. After that, you can either remove the DMSO directly by centrifugation or dilute the DMSO and change the medium the next day. Saying that, it is important to mention that freshly thawed cells are sensitive to shear forces, so too harsh pipetting should be avoided. In general, it is a good advice not to work with too many samples at a time and be organized. For example, label cryotubes or culture vessels first before you start to prepare the cells for freezing or thawing. I want to go on with some general remarks about cell banking, because having high-quality cell stocks is your backup system and the prerequisite for reliable research. Maybe the most important point is that you make sure that you don't freeze contaminated cells and that the cell line is really the cell line you think it is. So authentication of cell lines should be performed regularly, especially when you got the cells from another lab and when you use them in long-term or subsequent studies. The cells you're working with should also be tested for Mycoplasma regularly. Filter tips give extra safety, so they should be considered at least for such important steps like cell banking. The routine use of antibiotics is not recommended because they can mask a contamination with partially resistant bacteria. And please keep in mind that most antibiotics are not effective against Mycoplasma anyway. A two-stage cell banking system with a master and a working stock provides you constantly with early passages from your cell lines. A detailed record of the cell stocks is as important as clear labeling of cryotubes. No matter what you do with a cell culture, freezing, subculturing, or seeding cells for an experiment, it is important to determine the right time point for a particular cell type. In this image on the right, you see a typical cell growth curve of cells in culture. It is important to not let the cells overgrow until they are in the stationary phase and do not proliferate anymore. The optimal time point is when the cells are in their exponential growth phase, also called log phase, and this is when they are between 70 and 80% confluent. Plotting a cell growth curve and determining the population doubling time for a cell line is critical for understanding the optimal seeding density and the optimal cell concentration range for the routine subculturing of a cell line. When you launch an experiment, it is important that you can be sure that any changes in cellular behavior are a direct response to an experimental stimulus. For example, you want to test how the cells react to the treatment with a certain substance. So you add the substance to your cells and see increased cell death, or the proliferation rate slows down. To evaluate a direct correlation between the changes in your cell culture and the experiment you performed, a constant culture routine is important. A growth curve provides information about behavior during cultivation and thus facilitates the assessment whether any biological response can be attributed to the effect of a tested factor or whether it may reflect an imbalance within the culture system. Another factor that has often been described to influence cellular behavior is the passage number. The passage number is a record of the number of times a cell culture has been subcultivated. There are numerous studies giving evidence that the passage number affects a cell line's characteristics over time. It has been described that cell lines at high passage numbers experience alterations in morphology, response to stimuli, growth rates, protein expression, transfection efficiency, and more. So looking at all this passage number effects in different cell types described in the literature, the basic question one can think of is, how many passages are too many? And before we go on, I want to hand over this question to you and start our next poll. So, what do you think? How many passages can a cell line be cultured for? And the answers you can click on are not more than 20 passages, 20 to 60 passages, up to 100 passages, or you can click on the passage number has no influence on the cells. So again, I will wait a few seconds until the answers are coming in. All right, and now I'm going to show the results to you. So about half of you says not more than 20 passages. 36% says 20 to 60 passages is okay. Only 10% say passage number up to 100 is okay. And almost 4% of you don't think that the passage number has an influence on the cells. So in general, we can say keep the passage number as low as possible. Especially primary cells that have a finite lifespan are prone to genotypic and phenotypic changes as they adapt to in vitro culture. After a number of population doublings, they will stop growing. In contrast, continuous cell lines like cancer cell lines have an unlimited lifespan. But although they can proliferate indefinitely, also their genotype and phenotype can change over time. A lot of the well-established and well-characterized cell lines that are used in laboratories worldwide have been isolated from their original tissues decades ago. When you work with a continuous cell line that has been isolated from its original tissue 40 or 50 years ago, it is most likely not possible to obtain early passages from the cell lines. But you have to set a starting point as a reference to be able to obtain reliable results with this cell line. First of all, this starting point should be cells of high quality. The best way to ensure this high quality is obtaining cells from established cell banks like the ATCC in the United States, the ECACC in the UK, or the German DSMZ, just to name a few. You get well-documented, authenticated cell lines which serve as the best reference cultures for your experiments. By establishing a two-stage cell banking systems, like I showed you in one of the previous slides, with a master and a working stock, you have a constant supply of early passages from your cell lines. Now you can determine the safe passage number for your cell line of interest by establishing certain baselines. For example, test the cells for the presence and expression level of a certain protein. Monitor the proliferation rate over time. Keep an eye on any changes in cellular morphology. By getting to know your cells, you will get an idea of whether and how the cells change in relation to prolonged passaging. And maybe a very simple advice: do not keep cells in culture that are not actively used for experiments. So don't passage continuously, but rather establish new cells from frozen stocks if required. I already mentioned it. It is important that you get to know your cells. No matter which step you perform, first take a closer look with the microscope, not only to check the cells for possible contamination, but also to review the status of your culture. There's a lot of things beside contamination and confluency that can be checked with a simple bright field microscope. Especially in round-shaped culture vessels like dishes and multi-well plates, cells tend to accumulate in the center instead of spreading evenly across the entire surface when seeded into the vessel. Moving the vessel in a slow figure 8 or north, south, east, west motions after the cells have been added can help to disperse them evenly. But especially in small diameter wells, the movement of the liquid is not sufficient to distribute the cells. Here it helps when you dilute the cells to the desired concentration in a tube instead of pre-filling the wells with media and then add the required cell number. Changes in cell morphology, such as increased granularity, especially those observed around the nucleus, or vacuolation of the cytoplasm, can be indicators of an unhealthy culture. In some cases, it simply requires a change of medium, whereas in other cases, a more serious problem may be present, such as microbial contamination, senescence of the cell line, or inadequate medium or serum. As I already mentioned, cell culture media tend to build up a lot of foam. Air bubbles easily form during pipetting steps. When seeding cells, air bubbles can hinder cell attachment and should therefore be avoided. When cells die, they will take on a round appearance, detach from the surface, and float in the medium. Increased cell loss due to apoptosis or necrosis is a clear sign of an unhealthy culture that should be replaced by starting a fresh culture from frozen stock. As thawing is always a stress factor for the cells, it is normal to observe a number of dead cells floating in the medium of a recently thawed culture. A medium change should be performed 1 day after thawing to avoid accumulation of toxic products from dead cells in the medium. Proliferating cells are a sign of a healthy culture. When cells undergo mitosis, they assume a round shape and partly detach from the surface before they segregate into two daughter cells that will subsequently adhere to the surface and assume their characteristic morphology again. As mentioned before, when cell density is too high, cells will stop proliferating. For this reason, passaging at 70 to 80% confluence is recommended. Constantly monitoring the cells is crucial to reveal possible contaminations at early stages and avoid spreading of the contaminations throughout the lab. For microbial contaminations like fungi, yeast, and bacteria, a standard bright field microscope is sufficient to discover a contamination. In a lot of cases, they are even visible with the naked eye as color changes and turbidity of the medium appears. There is one type of bacteria that is not so easy to detect, though. Mycoplasma are too small to be seen in a standard microscope, and they don't visibly change the culture medium. Therefore, cultures need to be tested for Mycoplasma regularly. When a contamination with Mycoplasma or any other type of contamination goes undetected, it endangers the reliability and reproducibility of experimental data. As I already mentioned, cell cultures need to be tested for Mycoplasma on a regular basis. If they are tested positive for Mycoplasma, the infected cultures should be discarded immediately to avoid spreading the infection. When you get cultures from another lab, or when the status of a cell culture is not known because you have to wait for the test results, retain the potentially infected cultures in quarantine, for example, in a separate CO2 incubator, or if you have the possibility, also in a separate lab. There are different ways to test for Mycoplasma. Every method has its advantages and disadvantages. The best test method for you depends on different factors, like the access to the required equipment, the number of samples you need to test, and the time you can wait for the results of the test. On our new website, www.eppendorf.com/cellexperts, you can download a PDF file with an overview table listing the different Mycoplasma test methods, their requirements, and their pros and cons. This hopefully helps you to choose the best test method for your lab. There are not many cell culture quality standards defined in detail and are thus performed differently between laboratories or sometimes even within the same laboratory. Usually everyone is treating their cells a bit different, and this is one of the reasons that contributes to non-reproducibility of experimental data. A constant and detailed documentation helps to ensure that a cell line is always handled the same way. The source and date of receipt should be recorded by quoting the original designation of the cell line. The accession number should be retained if the cell line was obtained from a cell bank. Especially when establishing a new cell line, a unique name prevents ambiguity with respect to other cell lines or biological resources. Also important is an overview of culture conditions and procedures, including information about medium composition, growth surface, thawing density, doubling time, etc. The culture conditions should be adapted for each individual cell line. Reference images of the cells at different densities help to recognize any unusual changes in cell morphology. Especially when new people come to the lab or when you start to work with a new cell line, these images help to get familiar with how the cell lines or how the cells are supposed to look like when the culture is healthy. Images with different cell densities show how the morphology can change with increasing confluence. In cell culture labs, it is common that different people take care about the cultures during the week. One day, person A is changing the medium, the next day, person B is splitting the cells. But even if you are the only person working with the cells, it is important to document everything you do with the cells. Passage number, seeding number, split ratio, as well as cell viability should be constantly documented. By the way, when cell viability drops below 80%, it is recommended to reinitiate new cells from frozen stock to make sure that you cultivate a healthy culture. So taken together, implementation of standardized laboratory practice protocols contributes to the establishment of comparable parameters for higher reproducibility, independent of the person performing the experiment. You can create such a cell profile for every cell type you're working with, and every colleague who is working with the same cultures should have access to it. On eppendorf.com/cellexperts, we provide a PDF template for download, which you can print out and use in your cell culture lab. Apart from any user-dependent variations in handling the cells, there is another source of variability that cannot be overlooked when we are talking about reproducibility in cell culture. Animal serum is a standard supplement of cell culture media. The most commonly used serum is fetal bovine serum, also known as fetal calf serum, or FCS. It is added to the culture medium at a concentration usually of 10 to 20%. It contains vital nutrients, hormones, and growth factors which stimulate cell growth. Other serum components serve as binding proteins, which promote cell adhesion in vitro. Although the use of serum is well established in cell culture, it remains the most undefined component of culture media. Serum batches usually show qualitative variations. and the lack of uniformity in composition introduces high lot-to-lot variability. The development of serum-free media and serum substitutes is increasing. A variety of chemically defined serum-free media and synthetic serum substitutes is available from different suppliers. Despite of the ethical issues that come along with the use of animal-derived substances, for some applications it is crucial to have complete animal-free culture conditions. One example are bioprocess applications, such as the production of therapeutic proteins or vaccines. Also for the cultivation of stem cells, researchers want to resign the use of serum because stem cells are known to react very sensitive upon the slightest variation in culture conditions. However, there is not yet an alternative for all cell types available. In addition, the alternatives to FBS that are available are usually very costly. So there are a few things that you might consider to avoid the variability of serum affecting cell-based experiments. It is recommended to test different serum batches for the ability to support growth of a certain cell type and to stock up on a suitable batch. It is also possible to request that serum suppliers reserve appropriate volumes of a suitable batch. When the batch is used up, testing should be repeated to identify the next suitable batch. And I want to point out here that when you test a new batch of serum and find it to increase the growth of your cells significantly, it is not necessarily beneficial for the reproducibility of previous results you obtained with these cells. You should not simply screen for the lot that make the cells grow fastest. As I already mentioned, for finding a safe passage number for your intended study, testing of different serum batches should be tailored for your intended use. For example, have a look at the expression of certain marker genes of interest and monitor cell morphology with different serum batches. The last important point here is transparency. When you report how you screen or test serum, you enable others to reproduce the data more easily. And with this, I come to the last chapter of this webinar, misidentified cell lines. A misidentified cell line develops from a cross-contamination of two different cell lines, and usually this happens by an accident somewhere during the handling of cells. So it is a mistake in aseptic handling. For example, you are supposed to trypsinize one cell line after the other. But since you are a little bit in a hurry, you handle another faster growing cell line, depicted here in this image in pink, at the same time. By accident, a little droplet of the more rapidly growing cell line gets into the cultures shown here in green, and because the pink cells are growing faster than the green ones, with the following steps of cell culture, the pink cells become more numerous and start to overgrow the green culture. After a given number of subcultivations, you will end up with a dish of pink cells or a mixture of both. So, for example, if the green cells here would be Vero cells and the pink ones would be HeLa cells, you would end up with a dish that is labeled Vero but actually contains HeLa cells or a mixture of both. As soon as two cell types are mixed up, it is very hard, if not impossible, to distinguish those two. This process goes fast and it might go undetected, especially if the morphology of the cells is not drastically different. But although the phenomenon of cross-contaminated and misidentified cell line has been recognized for half a century, still to date we know that 15 to 20% of cell lines used are either cross-contaminated or misidentified. Currently, there are about 500 cell lines known to be cross-contaminated or misidentified. Misidentified or cross-contaminated cell lines are a big contributor to non-reproducible research. Therefore, it's important to check the identity of the cell lines you are working with. For human cell lines, identity testing can be performed using short tandem repeat profiling, or STR profiling. The analysis of short tandem repeats has become the standard for intraspecies identity testing of human cell lines. STR profiling can be performed in-house only if the required equipment is available. Adequate interpretation of the data requires high expertise and routine, so it can be more advisable to use a service provider for cell authentication. The development of markers for authentication of non-human cell lines is still an ongoing process, and more reliable polymeric STR markers which can provide intraspecies discrimination have to be developed. or identified. Several different methods like karyotyping, isoenzyme profiling, and DNA barcoding are available to detect interspecies and intraspecies cross-contamination in cell culture. You can find more information about the different methods for authentication of cell lines on our website. In addition, there is an upcoming webinar on June 29th where my colleague Dr. Nadine Melius will talk about the topic of cell line identity and how to prevent cell misidentification in more detail. Just have a look at our website and go to the trainings and webinar section where you can register for the webinar. Talking about training and education in cell culture, this is maybe one of the most important measures when you want to improve the overall reproducibility in your lab. We all know the cases where protocols and methodology have been developed by a student or a postdoc who is no longer with the lab. Someone who is new to the lab now has to rely on maybe not so perfect documentation and sets his own standard protocols, which again leads to the situation that everyone is doing it a little bit different. So one way that labs could get away from variability is having some expertise that stays in that laboratory. There should be at least 1 or 2 dedicated cell culture experts who are responsible for teaching good cell culture practice to the new colleagues and to keep an eye on proper documentation and standard operating procedures. This way, the knowledge of how to do things correct and how to set certain quality standards is resident in the lab, and this helps to improve long-term reproducibility. I'm almost at the end of the presentation. Again, I want to point out our new website, eppendorf.com/cellexperts, where you can find a lot of information about cell culture and the common drawbacks when it comes to working with eukaryotic cells, and some tips and tricks how to solve common issues in the cell culture lab. And with this, I want to start our last poll question and ask you for some feedback of today's presentation. So how do you rate the content of this webinar? Very good, good, okay, or poor? And again, I will give you a couple of seconds for answering the question. Okay, so thank you for your feedback, and thank you all for your attention for today's presentation so far. And yeah, we will start the question and answer session in a couple of seconds.
Yes, yeah. Okay, thank you very much, Jessica. I'm sure you'll all agree that there were some excellent points raised during the presentation. And as Jessica said, we will now move on to the Q&A session. We've had a number of questions in, so the first one for you is, how often should cells be tested for Mycoplasma?
There is no black and white answer to this question. So it depends on the lab you're working in. How many people are working there? How many trainees do you have in the lab? How many people that are not so familiar with aseptic techniques? So I would recommend to start with maybe every 4 weeks, and then you see what comes out of the testing. And if you have ongoing or subsequent Mycoplasma infections, then I would increase the frequency of testing. So I would start with a high frequency, maybe even every 2 weeks to every 4 weeks. And yeah, if you see that you have negative tests always, then you can decrease the frequency again. And yeah, as I said, keep an eye on aseptic techniques, that everyone is following certain standards to keep the risk as low as possible.
Okay, thank you, Jessica. The next question we've had is, does the use of antibiotics in cell culture media influence reproducibility?
Ideally, antibiotics used for cell culture should eradicate 100% of all contaminants. They should be non-cytotoxic for the cultured cells, and they should not interfere with experiments. In fact, none of the available antibiotics meet the mentioned criteria, and this is often overlooked. In addition, the usage of antibiotics varies greatly. So different researchers use different types and different concentration of antibiotics for the same cell types. Sometimes people are not aware that antibiotics are not stable for more than a few days at 37 degrees. So the concentration in the media bottle is decreasing every time it is heated and cooled down again. Some research don't add any antibiotics to their cell culture. So taken together, in most cases, the data that are published for a certain cell line or cell type were generated under non-standardized conditions. And this, of course, can be problematic when it comes to reproducibility of experimental data. So, yeah, definitely. I think the use of antibiotics does have an influence on reproducibility.
Okay, excellent. Thanks, Jessica. Moving on, the next question is, should I centrifuge the cells right after thawing them?
This actually depends on the on the cell type you're working with. So there are two ways of handling freshly thawed cells. Either you centrifuge the cells to get rid of the DMSO that you used for freezing, then resuspend the cells in fresh culture medium and put the cells in the culture vessel. The other way is to skip the centrifugation and only dilute the DMSO with fresh culture medium and then change the medium the next day. It really depends on the cell type. So there are cell types that are more sensitive to the centrifugation. So centrifugation of the cells when they are freshly thawed might be a problem. There are other cells that are more sensitive to the DMSO, even at low concentrations in the medium. So if you are not sure, I would recommend to simply test it. Take 2 cryotubes out of the liquid nitrogen tank. Thaw them. One you do a centrifugation step with. The other one is just diluted and directly put into the vessel. And then a few days later, you can check the viability of the cells. No matter which way you choose for the cells you're working with, I would definitely recommend to change the culture medium the next day because, as I mentioned during the presentation, thawing and freezing is a stress factor for the cells, so there will be dead cells in the medium after thawing, and toxic products will accumulate. So no matter if you do the centrifugation step or if you skip it, definitely change the media to get the next day to get rid of all the dead cells in the medium.
Okay, thank you, Jessica. The next question is, in our lab, different people take care of the routine steps like changing media and splitting. Everyone is doing things a bit differently, so how can we make sure that the cultures are always treated the same, independent of the person who's handling them?
Thank you. Yes, so this is what I wanted to point out in the presentation. You should agree to one standard protocol for the cell line. So such a cell profile that I introduced to you helps so that everyone knows how the cell needs to be handled. So it is important to agree in one lab for one standard procedure. So that everyone has access to information about a certain cell line, especially when people are new to the lab, or when people start to work with a cell line that they did not know before. It is important to have information that everyone has access to. So such a cell profile and the establishment of standard operating procedures that everyone follows helps to reduce the user-dependent variation that you otherwise would have.
Okay, excellent. Thanks, Jessica. So moving on, the next question is, how fast can a Mycoplasma contamination take over Vero cell culture and kill it?
Well, that depends a little bit on the feeding schedule that you have. If you, for instance, work with stem cells, you have to feed them, or you have to change the medium every day. So every time you change the medium. You also feed the Mycoplasma, and but the Mycoplasma infection is not so severe because you have new medium and therefore new nutrients for the cells, for the Mycoplasma cells. So it really depends on the type of medium and the cell. So it's not really easy to answer. So you might have a Mycoplasma infection without seeing any effect on the cell line. So this is why it is so important to do the regular testing, because it might completely go undetected. The cells proliferate how they used to. The medium does not change, and the cells look completely healthy, but you can still have a Mycoplasma contamination. And just because the culture look healthy, it doesn't mean that the Mycoplasma does not have any influence on the cells. So the metabolism of the cells might be affected by the Mycoplasma infection. So the regular testing and screening for Mycoplasma is very important.
Okay, excellent. Thanks, Jessica. We've got another question, which is, how can I check for a contamination from bacteria? We have black spots in the medium, but do not grow in LB medium.
These black spots in culture media are often an often seen phenomenon. If it is bacteria, you will soon see an increase if you work without antibiotics. So the bacteria will overgrow the culture within 1 or 2 days, very, very fast. But only if you do not have antibiotics that keep the bacteria that may be partially resistant on a low level. So the other thing is that most bacteria show active movement. So if you have a look under the microscope, fix one of these black spots that you see with your eyes and try to follow it. So if you see a directed movement, it is most likely a bacterium. If you only see that the black spots, are fidgeting around, then it is more likely cell culture debris or any precipitate of the medium. There is, on the website I was talking about, eppendorf.com/cellexperts, we do have a video that is showing bacterial movement versus these little black spots that are cell debris and that are not a contamination. So you can have a look at it. If you don't find it on the website, please just let me know, then I will send you the direct link.
Okay, brilliant. Thanks, Jessica. The next question is, when you mention outer plate evaporation effect, do you mean that's with the 96 well plate lid closed or the lid opened?
I was talking about the evaporation effect with the lid closed, because usually you don't have the open lid in the incubator, of course. And there is evaporation because it is just a loose lid, because you have to allow gas exchange, of course. So it's the evaporation effect that I showed you here. The numbers of 15 to 20% were measured with the lid on the plate over a period of 5 days of incubation at 37 degrees, and with a humidified incubator. So the effect is there, although the lid is on, although the incubator is humidified. This evaporation effect plays a role.
Okay, brilliant. Thank you very much, Jessica. Unfortunately, that's all we have time for today, but I'd just like to remind everybody that any questions we didn't have time to answer now will be answered offline as soon as possible. And you can also continue to ask questions even when watching the webinar on demand, and Jessica will get back to you with an answer as soon as she can. The webinar will now be made available on demand, so if you have any friends or colleagues that you think may be interested in this webinar or any future events, please feel free to share the link. We'd like to thank you all again for your time.